if we could use the body's own immune system to attack cancer cells? One promising new method that has been developed are neoantigens, which prime a patient's immune system to fight the cancer on its own. But how can the immune system differentiate cancer cells from regular cells? They do so by looking for antigens key to an individual's tumor-specific mutation. Finding these can lead to immunotherapies specific to each person's cancer. Neoantigens are developed by finding mutations in the DNA that are present only in the cancer cell, and then developing new antigens that can bind and attack them. However, these mutations are incredibly difficult to find.
Next-generation sequencing made it possible to rapidly sequence someone's genome, but it generates terabytes of data that need to be parsed to find mutations of interest, which is where a lab comes in. We're from the FRI Big Data in Biology stream, and we're looking for mutations in cancer cells that lead to new antigen prediction. Particularly, we worked with 70 samples of multiple myeloma cell lines. However, looking for a couple of mutations in a seas of billions possibility continues to be a challenge. It's like a book Where's Waldo, where you have numerous regular people, but only one Waldo. Here is the mutations among the seas of sequences, or Waldo. Did you find it? Luckily, many tools have been produced to find these mutations. Unfortunately, these tools are really difficult to run and require a lot of computing power, especially when trying to run them across 70 samples. So we used the Texas Advanced Computing Center to run these tasks on dozens of computers at the same time. The work would have taken about 11 months to complete without it. But these tools vary in efficiency and accuracy.
We looked at 4 standard tools and then compared the mutations we found to ones that have already been found by other researchers. By assessing metrics such as sensitivity, specificity, F-score, and runtime for each tool, we found that VARScan performed definitively better. In the end, we found about 1,400 expressed mutations. We discovered which tools should be used to identify mutations in cancer sequencing data and what expressed mutations could be used to identify and target these specific cancer cells. This brings us one step closer to being able to sequence a cancer patient's genome and design therapeutics that are targeted to their specific cancer cells.